Chapter 4, Nucleic Acids and the RNA World. This chapter discusses what is a nucleic acid. We'll look at DNA structure and function, RNA structure and function, and we'll look at the stability and versatility of both of these molecules. And at the very end, we'll talk about how could life have formed, and really, was it an RNA world first? So what does DNA represent? It represents deoxyribonucleic acid, and it stores genetic information in eukaryotic and prokaryotic cells. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And many researchers think that RNA came first before DNA, and this is called the RNA world hypothesis. And as you can guess, this will be important for your next exam. So what they propose is that RNA both stored genetic information and it catalyzed its own replication. And so this was the beginning of life itself. In other words, RNA is considered a naked self-replicator. Okay, so what is a nucleic acid? Well, a nucleic acid is another macromolecule, just like a protein is a macromolecule. And another word for macromolecule is polymer. So all polymers are made up of monomers, that one single building block. And the monomer for nucleic acids are nucleotides. Nucleotides are made of three components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. Both the phosphate group and the nitrogenous base are bounded to the sugar molecule. So in your book they distinguish between ribonucleotides and deoxyribonucleotides. But the truth is that most of the time we just simply say nucleotides. But what is the difference between the two? Ribonucleotides have a sugar called ribose, whereas deoxyribonucleotides have deoxyribose. Well, what does deoxy mean? It means lacking oxygen. So at the 2' carbon, which we'll discuss in a little bit, that 2' carbon, instead of having an OH group like ribonucleotides have, it only has a hydrogen. So that is the big difference between the two. Believe it or not, that missing oxygen makes DNA a much more stable molecule than RNA. So if we were to take a look at the nucleic acids, we can also group them according to the types of nitrogenous bases. So remember, there's 3 parts to a nucleotide: a sugar, a phosphate, and a nitrogenous base. Well, there's 4 types of nitrogenous bases. So I always think about it, I use the mnemonic device: apple, tree, chewing gum. Adenine, thymine, cytosine, and guanine. And then there's uracil, and uracil is only found in RNA. So besides that, you're going to have to know the difference between purines and pyrimidines. Purines are 2 ring structures, and they compose of adenine and guanine, and pyrimidines are 1 ring structures, and it's cytosine and thymine. So there's some mnemonic devices that you can use to remember that. So one is for purines: pure as gold, pure as gold. So that reminds you that purines consist of adenine and guanine. And pyrimidines, the mnemonic device is cut the pie, cut the pie. And that will remind you that cytosine and thymine are part of pyrimidines. In figure 4.1, you can see in part A, the nucleotide, the 3 parts of a nucleotide: the phosphate group, the 5-carbon sugar, and the nitrogenous base. If we look at the 5-carbon sugar indicated by the red molecule, we see that at the corners we have 1, 2, 3, 4, and 5 prime. And notice that it's in a clockwise fashion. At each in the corners, it represents a carbon. And so this is how the carbons are labeled. Now if you look at part B, the sugars, you can definitely see the differences in sugars between RNA and DNA. Notice that ribose has an OH group at the 2' carbon, whereas deoxyribose only has a hydrogen connected to the 2' carbon. And then in part C, when we look at the nitrogenous bases, you can see the differences between pyrimidines and purines. Purines are singled ring structures and purines are doubled ring structures. And remember, cut the pie, pure as gold. Could chemical evolution produce nucleotides? So studies show that we cannot actually produce nucleotides, but we can produce the sugars and the nitrogenous bases that are found in nucleotides. And in deep sea vents, it's very easy to produce ribose, or you see large concentrations of ribose. Okay, so what connects one nucleotide to another? So when we look back at proteins, isn't it true that the peptide bond connected one amino acid to another? So now that we're looking at nucleic acids, the phosphodiester linkage links one nucleotide to another. And it says here that the phosphate group on the 5' carbon of one nucleotide is connected to the hydroxyl group of the 3' carbon of another. Now that's sort of hard to imagine, but we'll see in the diagram how they're connected to each other. So it says here the 2 types of nucleotides are involved. So remember, ribonucleotides for RNA and deoxyribonucleotides for DNA. I love this diagram, figure 4.2, and what it shows is that between one phosphate group and a sugar that we're going to have a condensation reaction. Maybe another name for condensation reaction is dehydration synthesis. And remember, water is removed to form the bond, and the bond for nucleic acids is the phosphodiester linkage. So you see on exactly the left-hand side, or the right-hand side, excuse me, that water is removed. DNA and RNA strands are also directional. So it says here that the phosphodiester linkages form a sugar phosphate backbone. So it's, if you think of the double helix as a ladder, then sugar and phosphate make the sides of the ladder, and the steps are made out of nitrogenous bases. So what causes the directionality? One end of DNA ends with an unlinked 5' phosphate group. The other end ends with an unlinked hydroxyl group at the 3' carbon. And we'll see this in just a second. So how do we express this directionality? We call it the 5 to 3' direction. And this is going to be important later on when we talk about transcription and translation. In this diagram here, you can see at the very top of the DNA molecule that the phosphate group ends at the 5' end. So if you look closely at the sugar, you'll notice that the phosphate is connected to the 5th carbon. And if you look at the opposite end at the bottom, you're going to notice that at the very end, the 3' carbon, you see the OH group. So this gives it its directionality, 5 to 3'. So how is it that a DNA molecule is made? Well, a DNA molecule is pretty stable, and it's not a self-replicator like RNA, so it definitely needs enzymes. And as you guys know, enzymes are a type of protein, and this is used to catalyze the polymerization of DNA. Keep in mind, polymerization just means putting monomers together to create a polymer. Okay, so it says here, phosphate groups are negatively charged and they repel each other. But the truth is where does the energy come from? And it actually comes from this repulsion of negatively charged oxygens found in the phosphate group. So those bonds that are holding these negative charges should be very strong. So when they're broken, it releases energy to build from a small building block, a monomer, to a polymer. So actually activated nucleotides are the building blocks of DNA and RNA. One famous one, the most famous one of all of the activated nucleotides, is ATP, adenosine triphosphate. In this diagram 4.4, if we look at ATP is an example of an activated nucleotide. You see that triphosphate tail. And when you look at the triphosphate tail, you see all of those oxygens with a negative charge. But you guys know that like charges repel each other. So those bonds holding the oxygen and phosphorus together must be mighty strong to hold them together with those charges repelling each other. So if we break off two of those phosphates from the phosphate tail, when we break that bond, a lot of energy is released. How much energy is released? It's exactly 10.9 kilocalories per mole. And that's enough energy to build a DNA molecule. So the primary structure of DNA is actually the sequence of nucleotides. And doesn't that sort of remind you of proteins? The primary structure of proteins was the sequence of amino acids. So that's pretty much same old, same old. But what is the secondary structure of DNA? The secondary structure of DNA is the double helix. And this double helix starts with complementary base pairing. So back in the day, when they didn't exactly know what the DNA molecule looked like, we had this thing called Chargaff's rule. And Chargaff's rule was that the number of purines was always equal to the number of pyrimidines. So remember, pure as gold and cut the pie. So when we say that, that means that there's an equal number of T's and A's and an equal number of C's and G's. And that's because purines equal to the number of pyrimidines, and we'll see that this is the beginning of complementary base pairing. The other clue to the secondary structure of DNA was from X-ray crystallography. And Rosalind Franklin has been given the Nobel Prize after she passed away because actually her work was taken from her without her knowledge, and you could see the twisting nature of the double helix. And from there they knew the spacing and they knew exactly what the molecule looked like. Okay, so Watson and Crick are given credit for the structure of DNA and for actually understanding complementary base pairing. A always complements T and C always complements G. So that goes back to my mnemonic device, apple, tree, chewing gum. A always pairs with T and C always pairs with G. And the other thing that we need to know in the secondary structure is that the strands are anti-parallel. So one strand runs from 3' to 5' and the other strand runs in the opposite direction, 5 to 3. So DNA forms a double helix, and that's the secondary structure. It says here that the sugar and phosphate backbone faces the outside and the nitrogenous bases face the inside. They're like steps to a ladder. In this diagram here, we see that purines equal pyrimidines. In other words, a double ring is always matched with a single ring. Why is that? If you have two double rings together, they're crowded, they're overlapping. If you have two single rings together, then they're spaced too far apart. So it's sort of like Goldilocks and the Three Bears. A double ring and a single ring structure together is just right. So you see in part B that hydrogen bonds are actually connecting the nitrogenous bases from each of the strands. So hydrogen bonds are not just used to connect water molecules together. They're not just used in proteins for alpha and beta helices, but they're also used to actually connect nitrogenous bases. And you can see this in part B. And in the last part, you see the anti-parallel strands. Now, when I look at anti-parallel strands, I focus on the sugar molecule. Notice the sugar molecule is pointing up in one of the strands, and in the second strand it's pointing down. And this, to me, automatically indicates that they're anti-parallel strands. It says here that one turn of the helix occurs every 10 bases. You won't have to know this for the exam, but it's sort of information that's nice to know. And as it twists, you know, I always think of Twizzlers. So it's twisting, but the twists aren't equal. So there's a tight twist and then a loose twist. Another way of saying it is that there's a major groove and a minor groove. If we look at this diagram, you can see that there's wide spacing alternating with small spacing and narrow spacing, and this is called major and minor grooves. I also like this diagram because you see that nitrogenous bases, apple tree chewing gum, so you can see complementary base pairing and you can see anti-parallel strands. Here's a stick model representation of a DNA molecule. Here's a ball model of a DNA molecule. Okay, so what's the next structure? We have primary, we have secondary, and next is the tertiary structure. So it's the third level of structure. So what is the third level of structure? Well, normally DNA looks like strung out spaghetti, or maybe it looks like vermicelli. It's very thin and it looks just nice and stringy inside of the nucleus. But it's when it's ready to replicate, it condenses and it begins to bind tightly upon itself. In other words, it forms supercoils. And when it forms supercoils, that is the tertiary structure of DNA. And to form these supercoils, it's wrapping itself around proteins. You know, like thread is wrapped around a spool. So it becomes more compact, and when it becomes more compact, that means that the cell is ready to divide, either for mitosis or meiosis, and we'll talk about that later on. What is the function of DNA? DNA is definitely the information molecule. So DNA stores and transmits biological information. And this information is the program for an organism to grow and reproduce. And its language is in the bases. I always think of the movie The Matrix. Remember where actually environments were made with a binary code, like a streaming cascade of zeros and ones. But in this case, instead of zeros and ones, we have A, T, C's, and G's. So the order of these nitrogenous bases is what actually codes for us and makes us us. So DNA's primary structure serves as a template for the synthesis of a complementary strand. So when DNA is replicated, how does it replicate? Well, everything boils down to the complementary base pairing. So a new strand is actually made of one old strand and one new strand. And keep in mind that DNA can't replicate by itself. It needs an enzyme to help build the new strand. So what are the basic steps to building a new strand of DNA? First, the two strands that are bound together in a double helix, they have to unwind. And then they're separated. So the hydrogen bonds holding them together are usually broken by applying heat. So next you have a template strand and a complementary strand. And that template strand can be used to build a new strand, because we know that A always complements T and C always complements G. In this diagram 4.7, you can see the three basic steps. The first step, the double helix is unwound and heat is applied to break the hydrogen bonds between the nitrogenous bases. Now we have two template strands and two new strands are being built, and now we end up with two new strands of DNA. But each of these strands is composed of one old strand and one new strand of DNA. Okay, so what can we say about the DNA double helix? Well, first of all, it's very stable and it's resistant to chemical degradation, and it's a very reliable storage for genetic information. So what is this like? It's sort of like our social security card, our driver's license, our credit cards. Those are the important cards that make us us. And we always store it in our wallet or in a very safe place at home, and we don't give it out to people because we know that that is what identifies us as us. The same thing is true at the cellular level. DNA has to be a stable molecule because it is a reflection of what makes us us. So it says here, stable molecules such as DNA are very poor catalysts. So remember what a catalyst is? A catalyst is something that speeds up a reaction. So DNA cannot build itself. It actually needs an enzyme to help build the complementary strand. So for this reason, many biologists believe that DNA was not the very first molecule because it's too stable. The very first molecule must have been RNA. It stores information, but it also acts like a protein. It can self-replicate itself. It doesn't need an enzyme. RNA, like DNA, has a primary structure, and that primary structure is the order of its nucleotides. And just like DNA, it has a sugar phosphate backbone, and then the steps to it are the nitrogenous bases. But keep in mind that DNA is double-stranded. On the other hand, RNA is a single strand. The other major difference between the two is that RNA contains ribose as a sugar and DNA contains deoxyribose. So that is the second difference between the two. What's the third difference? The third difference is that instead of containing thymine, as DNA does as one of the four nitrogenous bases, RNA contains uracil. The secondary structure of RNA is a little bit different from that of DNA. So RNA can form this thing called a hairpin structure, and that means that RNA can fold on itself, and when it does, it says here that the nitrogenous bases start aligning with each other, and it almost looks as if they're forming an anti-parallel double strand, something that looks akin to DNA. So we have the RNA hairpin structure for a secondary structure. So in this diagram you can see that there's a loop, and that's why it looks like a hairpin, and then you can see that as the arms fold on each other that there's complementary base pairing. It says here that RNA can also function as a catalytic molecule. So it says RNA molecules can have a tertiary structure, and that means they can start folding on each other and they can form complex shapes. And I bet you're thinking, that might look like something like a protein. And the truth is, yes, it can. So RNA is much more diverse in size, shape, and reactivity than DNA. RNA is much more reactive than DNA, and RNA is much more versatile. So it's an information molecule, but it's capable of self-replication, and it can catalyze its own reactions. And there's a special term for that you have to know for the test, and this is called ribozyme. When I think of ribozyme, I think of RoboCop. You know the movie RoboCop? It's the cop that almost died and they made him half cop, half robot. Well, that's what a ribozyme is. It's half information molecule and half protein, and together it's a self-replicating information molecule, ribozyme. And then in this drawing here, you can see that RNA is folded on itself, forming this complex structure. And it has like this active site. And this is where it can actually catalyze its own reaction and replicate itself. I love this summary table 4.1. It compares both DNA, its primary, secondary, and tertiary levels of structure. Please remember to look at this, and please know this for the next exam. Again, RNA is capable of self-replication, and that's very important because it doesn't have to rely on other enzymes or proteins to help build a new molecule of RNA. And here it shows exactly how that works. So here it is catalyzing another strand of RNA. And notice it's doing that using complementary base pairing. The first life form. So what makes the first life form? A naked self-replicator. And what is the naked self-replicator? The naked self-replicator is RNA itself. So RNA, when we talk about an RNA world, we're talking about a pivotal moment. When RNA evolved, this was the beginning of life itself. So when we had RNA, then life was ready to begin because it carries information and it's able to actually copy itself. So most researchers believe that when we saw RNA, it was a pivotal moment in the formation of life, and this was the beginning or the starting point of having cells. There's additional evidence that shows that most ribozymes aid in protein production, and they must have evolved first before proteins themselves. So this is another indication of an RNA world hypothesis. And why is this so important? Because RNA has three characteristics of life: information processing, replication of heredity information, and evolution by random changes. So it carries three of the five characteristics of life.